everyone. Today I'm going to give you the lecture on immune response kinetics and memory. This would be the lecture for our class on November 8th, 2023. Though because I am still out of town, I'm giving this to you as another video lecture. Just a couple of announcements, which I will also mention by email. On our schedule, it says that we're going to be discussing a primary lit paper on Friday. I think that I would rather talk about our next topic on Friday, which is NK cells. And we will then discuss a paper likely about immune response kinetics and memory sometime next week. Probably Monday, but I will give you more information about that in class on Friday. You'll also notice that next week you have an assignment called the practical immunology assignment due. That assignment will actually get pushed back until probably Friday of next week. So just know that those schedule changes are coming up on the horizon. There are a bunch of details that I want to talk about today that are related to immune response kinetics and memory. This is a tricky topic for me to talk about because during the SARS-CoV-2 pandemic, as we have been measuring many patients' immune responses, and as we have particularly been measuring some immune responses to vaccines, I think that there are a lot of details about immune response kinetics and memory that we've learned, or questions that we've actually been able to get some really cool data to address. That makes it tricky to pick a paper at this point because there are so many that I find so important. But it also means there's a lot of up-to-date things and a lot of places where I sometimes realize the things I've been telling you about some of these pieces in class might be a little tricky compared to where things actually are. Also, some of the details of immune memory are details that I worked on in my dissertation work as a graduate student. And so there are a million really picky details about this that I have spent a lot of time thinking about and have to kind of hold myself back on telling you about. So I apologize for having like weirdly strong feelings about a couple of slides that we're going to see today. So everything that we're really talking about today is based on things that we've already seen with regard to phases of a primary immune response. And you have previously seen some of this figure that is on the left before. So what you can see here is this plot that I've shown you before with size of a specific immune response. This is any adaptive immune response. It can be B cells. It can be antibody responses. It can be T cells, whichever one you would prefer on the y-axis. And you can see our primary immune response that we've been looking at in response to that microbe here with time on the x-axis. And you can see where one might get infected with a virus. And we'll see a little bit of a delay in setting up that adaptive response, during which time that virus can sort of replicate unchecked because we have not yet primed that innate response. That will also lead to some illness because of some of the damage that will be coming from that virus. And eventually our adaptive immune response will start to come up. And we've talked about some of the reasons for the delay in that response. And then eventually that primary immune response will actually come down. And you can see some of this in a figure from a different textbook on the right, where you can see, again, relative size on the y-axis, time on the x-axis, and you can see the amount of virus happening kind of early, and that virus coming down and being controlled after we've got this good CD4 and CD8 T cell response. So once we get this peak adaptive immune response, we're going to get rid of this virus. You can see the virus going down in black, or just stopping existing in my little figure. Then we feel better, and we all live happily ever after. You can also see a version of this same plot when you look at this figure from your textbook. So we can see that we'll have our first infection with some microbe, and there will be a delay before we have our adaptive immune response of both effector T cells and antibodies coming from plasma cells that will eventually clear that microbe. But what you can also note is that this figure from your textbook depicts a whole lot of other kinds of phases and tells us about some other things that are going on at different times following some of that immune response. And so those types of things are really the things that we're focusing on today. So here you can see some of the specific events happening in this phase that we're currently thinking about throughout this process. So we can see our naive cells becoming activated, as we've already seen. We can see clonal expansion, basically the division of these cells. Again, all of the events that we're seeing on this slide are events that are taking place in a primary lymphoid organ. We can then see differentiation of those cells to plasma cells or other types of effector T cells, say, like CTLs or helper T cells, and then we can eliminate the antigen. So all of these events that you're seeing are sort of the events we've been talking about over the last few class periods. So we have to talk a little bit about what happens after antigen elimination. And what happens after antigen elimination is related to some of the details that are related to what's going on with our effector cells that are made following differentiation of those cells. So we've talked about how our naive cells change and become these more effector type cells through this process of differentiation after they're activated. And we need to think a little bit more about what's going on with these cells, what exact properties they have gained, because that will help us start to understand some of the details that are going to happen after antigen elimination. So here we can start to see some of the differences between naive cells and effector cells. Both of the figures, actually all of the figures on this slide, as well as on the next slide, are specific to thinking about the difference between naive T cells and effector T cells, though we can come up with very similar compare and contrasts if we want to do the difference between naive B cells and effector B cells. I just don't have kind of the exact slides to show you some of those things. What you will notice is that on the left, we have a table showing us some different genes, how long it takes for those mRNAs to come on. But the really important thing that I want you to notice is this column that's all the way on the right in this table, which says ratio of activated to non-activated cells. So basically the idea that is being shown here is how much more of this transcript is present in a cell that is an effector cell or an activated cell compared to a naive cell. The idea that this is trying to show you is that there are certain genes that are made very strongly and at very high levels in effector cells that were not being made in naive cells. So for example, we can see that FOS, remember that was one of our transcription factors that was part of AP1, is made more than 100 times more in activated cells compared to non-activated cells. So you can imagine those effector cells, which were the activated cells, are going to be able to signal really well. We've got way more NFAT, 50 times as much NFAT. We have over 1,000 times as much IL-2. You can see also for many of these other types of cytokines, we have vast increases in the amounts made in effector cells compared to naive cells. You can also see that there are big differences in cyclins that are made in those effector cells compared to naive cells. And that can help the cell undergo cell cycle and proliferate more. You can also see a similar type of table from your textbook in the upper right. And so you can see a bunch of different proteins that are turned on very strongly in effector cells compared to naive cells. Many of those are things like interferon gamma or granzyme B, things that are going to help an effector cell make its effect and be able to act really strongly against a microbe. What I also like about this table from your textbook is you can see that there are some proteins that effector cells stop making compared to the naive cell. So for example, you can notice that CD62L, which is L-selectin, stops being made by that effector cell that wants to go to tissues because that protein is important for homing to a lymph node. And you can see that there are a few other changes. A particularly important change is a change in CD127. But one other change that I want to point out is this change in BCL2. Note that the effector cells have a good amount of BCL2, and there's quite a bit less in the effector cell. BCL2 is really important to help cells survive and to help cells avoid apoptosis. And so you can see there's this change in proteins involved in apoptosis. And that's one really important piece that we should be aware of with effector cells. Effector cells are really working hard in order to be able to deal with microbes. These cells are highly, highly specialized cells. They're making huge amounts of transcripts and then huge amounts of proteins, and they are working very diligently. They probably are going to have to, sacrifice some other particular cell functions. And effector cells are often known as being relatively short-lived, meaning that those cells are potentially quite prone to cell death. Sort of like, these cells are going to live hard and die young. And so we sometimes think about effector cells as being relatively short-lived effector cells, because once they've devoted all these resources to this ability to really act against a pathogen, those cells may not have the resources they need to live for a super long time. And the BCL2 data that you saw kind of hints at that. You can also see this in a case with B cells. Remember, our naive B cell has a B cell receptor, a surface immunoglobulin. It has class 2. It has the ability to proliferate. It has the ability to do somatic hypermutation and class switch. But a plasma cell, which is the effector cell of the B cell lineage, really just has one job, which is that high level of antibody secretion. And that cell has really, really dramatically changed its whole cell structure in order to make high levels of antibodies. And so that may not be a situation where that cell is going to be able to live for a super long time, because it has altered its biology quite a bit. And so we can kind of think about these effector cells. Again, they're going to be phenomenal at dealing with pathogens, but they are often thought of as being rather short-lived. We also are learning more and more about the fact that they may do slightly different forms of metabolism than naive cells. Metabolism has become a really hot area for many immunologists. So what you can notice here is that if we look at a naive cell, whether it's a CD8 cell, which is shown up here in the upper left, or a CD4 naive cell, kind of shown down here, is doing about half of its metabolism by the blue type of metabolism, which is oxidative phosphorylation. That's kind of the aerobic respiration types that you've learned about before. It's also true of the CD4 cell. You can see that the cell is also doing about half of its metabolism with the yellow type, which is known as fatty acid oxidation. And what you should notice is that as that cell becomes an effector T cell, whether it's a CD8 effector T cell or a different type of CD4 effector T cell, the cell changes its type of metabolism because that cell might need to get faster energy. It may need to do a super fast form of energy, maybe a form of energy production that's less efficient, but that will allow that cell to get energy fast. So the idea, again, is that that cell is sort of making sure that it can do really well at combating pathogens now, but may not be sort of setting itself up to do well long term. And so we can see some of this information here, or some of this sort of as a consequence here, that after those cells have sort of undergone some big expansion, the majority of those cells are going to die. And this is known as the contraction phase. There are a number of different reasons, as this figure on the left or on the right shows, why contraction could happen. But the key thing that you want to see here is that the peak level of our primary immune response, whether it's a B cell response or a T cell response, is going to go down over time once our antigen is gone. Notice that it does not go down to zero. We end up with more of a response than we had before. It wasn't 100% of those cells died. It was just a large majority of them. You can also see this in the upper right, where we can see our CD4 and CD8 T cells sort of reducing over time. But this contraction phase is really key. I don't think I originally understood quite how important the contraction phase was. And when we were doing some experiments when I was a graduate student, one of my colleagues spent a bunch of time trying to actually inhibit contraction. She had the idea that perhaps we could make vaccines better if we got rid of contraction. You know, we made this great immune response. Why let all those cells die? So let's try to block their death. And everything she did didn't work too well, because it turns out that contraction phase is actually incredibly important. And there are lots of redundant mechanisms making sure it happens because it is so important. And one way that you might think about this is that you can think about that swelling that happens in a lymph node as you're making that expansion of effector cells. You can imagine that lymph node's getting pretty full. We're going to be using a lot of resources towards that clone of all of those cells that are responding to that antigen. If you didn't get rid of some of those large number of expanded cells, you could imagine that it would be really hard to have the room in the lymph node, or the resources in terms of the glucose and ATP or whatever resources you want to think about, for the next clone, to expand the next time you get infected. You know, by this time in your life, you'd probably just be a walking lymph node if you had kept every single expanded effector cell that you've ever had in your entire life. So it's actually quite important that those effector cells die. And so we can see that we often will see this contraction phase. This contraction phase is often known as activation-induced cell death, or AICD. So when our cell is activated, it also will be sort of induced to be more short-lived and potentially undergo cell death. which is particularly important to allow for homeostasis of these responses. And so I show you this in both a cartoon form on the left, but if you look at this real response that I measured in mice on the right, you can easily see that contraction phase as well. One of the proteins that's really critical for this contraction phase is a protein that you've already heard about this semester, which is Fas. And so we've talked about the fact that Fas ligand can be on CTLs that are active, and that can interact with this protein Fas. When Fas ligand interacts with Fas, that induces signaling in the target cell so that target cell undergoes apoptosis. But it turns out that as a T cell gets activated, that T cell starts, or as any lymphocyte gets activated, that cell starts to make Fas so that it can also be killed easily by cells expressing Fas ligand, as sort of like making a little bit of a kill switch for that cell, so that cell cannot be produced for too long of a time. Sometimes we even see activated T cells that use their own Fas ligand on their surface, to interact with their own Fas molecules on the surface, and they sort of have to bend in this really weird way because that contraction is so important. If you don't believe me, this is actually a patient with an autoimmune disease called ALPS, or autoimmune lymphoproliferative syndrome. And what you can see is that this patient has permanently swollen lymph nodes because she has a mutation either in Fas or Fas ligand, so that she actually cannot do that contraction phase of those cells and doesn't have those cells undergoing contraction particularly well. So in fact, they are still staying there and filling up her lymph node. Patients with ALPS often have a lot of problems with autoimmunity and excess inflammation because those cells, you can sort of imagine when those cells don't have any more antigen to be attacking, they might start to, get up to some mischief and start to cause some other types of damage. Remember, those cells are all a little bit partially self-reactive. And so this is kind of this type of phenotype, and the autoimmune diseases that come with it are problems that happen if we don't have that nice contraction phase. So one reason why I think it's important to point this out is that this is a place where I think immunology has been in the news and has been an interesting thing for people to think about over time. And so these are some data looking at levels of antibody responses in people who were infected with SARS-CoV-2 following their infections. And so you can see we're looking at antibodies against two different antigens, and we're looking at patients who were either at 1.3, 6.2, or 12 months after their infection. And we can see that the antibody levels are going down. When data like this were observed, some people looked at this and really freaked out because they said, oh, immune responses to SARS-CoV-2 don't last very long. People might get reinfected frequently. Something weird with this. This is doom and gloom, and what's going to happen with vaccines, and oh my goodness. But when some immunologists looked at this, we said, oh, so it looks like there's a contraction phase of the antibody response, just like the textbook says there should be. So that's cool. There's been a lot of debate over whether or not that contraction is happening kind of with the right kinetics. Is it happening too fast? But the fact that we're seeing contraction really should not be alarming to people, and we can debate whether or not we're seeing the right contraction. I often find these two papers particularly interesting. These papers came out basically like right at the same time. They're both looking at the contraction of SARS-CoV-2 antibodies in humans. And you can look at just the data from these two figures, and you can kind of see they're showing about the same kind of curve for that contraction over time in these patients. And what you can also notice is that one of them talks about how robust the antibody responses are, and one of them talks about this terrible decline. And so, again, this is sort of a thing to debate, but realize that sometimes when people are talking about this decrease that we're seeing in antibody responses, the take-home message might be, well, yeah, cool, that's what ought to be happening. There are a number of other things that we can think about when we think about details of immune response kinetics. One of those things has to do with what will happen if we happen to be infected again with that pathogen. And observations about this have given us a fair amount of insight into some of these details about what happens when the antigen goes away. In the very short term, if you get infected again, perhaps you have an infection, you get over it, but your person that you live with, a family member, friend, whoever, is infected and you get re-exposed. Well, right after your microbe is cleared, you probably haven't yet had a full contraction phase. Your effector T cell levels, your antibody levels are still really high, and that may allow you to have your infection basically aborted and stopped in its tracks, so that the microbe cannot actually even establish itself in your body. But as time goes on, those effector cells, those effector responses will decline through the contraction phase. Those short-lived effector cells will decrease dramatically in number, and we'll have this sort of number of other cells. Again, note that it is increased from what we had at the beginning, that can persist. But what we also notice is that if sort of after that period of time you get infected again, you're going to see a big difference in the type of response that you make. And so what you'll notice is that this second infection happens over here. And one of the things that is really different is that we don't see the same kind of delay in the adaptive response that we saw after the first infection. You can also see that we have a much more dramatic increase in terms of numbers, in addition to the size of that response. And so we need to, again, talk a little bit about what's happening to allow that change in response to happen. You might wonder a little bit about some, again, practical consequences of some of the things that we are talking about here. And one of the practical consequences that I want to mention, I sort of got to this a little bit on the previous slide, is that the exact way that previous exposure can protect you can vary a bit over time. Early after infection, your level of antibodies will be very, very high. That might be because you're in this early phase where you haven't yet gone through a contraction phase and you still have a lot of effector cells present. That might mean that you have, you're able to block, say, transmission of some different microbe coming into your body. Later on, perhaps you have fewer antibodies and you have this change in cell type that we're going to be talking about, and that may not protect you from actually acquiring the microbe. The microbe might be able to get into your body, but you now might make a different and better response. And so just remember that many of these different types of immune responses that we see are changing over time. And so some of the details of what we see in an immune response over time can change. This also goes back to some of the information that I've told you a little bit about before. I will point out very quickly that the information that is on this slide is a dramatic oversimplification of something that I spent a lot of time thinking about in graduate school. I don't 100% agree with all of the ways that things are presented here, but we're going to go with it, even if it makes me feel a little weird. Because what you can see is this figure that you saw on the right, where we have our naive cells and we have this expansion of a large number of effector cells. And we saw that 95% of them will die due to contraction. But what you should also notice is that there is also this set of other cells that don't die, that survive. And those cells have a set of special properties. Though you can also kind of see, those larger number of remaining cells after this response. Those cells are called memory cells, and those cells are responsible for that improved response that we're going to see the second time around because of the unique properties that they have. So we can have effector cells and memory cells produced for both B cells and T cells. Again, on some of the next few slides, you're going to be seeing some examples that are either B cell examples or T cell examples. But the general idea of effector versus memory cells is something that we can see in both B cells and T cells. So remember, we either start with a naive T cell or a naive B cell. Note that that cell could potentially end up being an effector cell or a plasma cell, or that cell could become a memory T cell or a memory B cell. And one of the really unique features about memory responses is that those memory cells have become specialized, just like the effector cells had, but they have a different type of specialization. And one of the big features of a memory cell that is unique about it is that that memory cell is very long-lived. So unlike the effector cell that's short-lived and is sort of ready to undergo apoptosis after it does its job, that memory cell is going to be able to live for a long time or be able to divide and have some progeny that are going to sort of keep that lineage going for a very long time. There was a ridiculously cool paper that came out in earlier in 2023 that actually looked at some details of how long those memory cells can live, and we can talk about that a little bit later. But these are some data that people have talked about in terms of longevity of memory responses. Here you can see in this figure that individuals were looking at antibodies and are seeing antibodies that persist after more than 30 years after vaccination. Often we do these with vaccination because we have a defined time where we know you saw the antigen, and we can in often cases know that perhaps you have never actually been infected. Maybe it's a virus that isn't really around. And so we know how long it's been since you've seen the antigen. You haven't seen it again. And you can also see that CD4 and CD8 cells are lasting for long periods of time. In terms of relative magnitude, you can also see this on the left. If we look at antibody levels in unvaccinated people, we basically can't find them. They're below the limit of detection. And vaccinated individuals have antibodies that we can detect for more than 70 years after they've been vaccinated. Similarly, if you look at CD4 and CD8 T cells, we can see in some patients those CD4 and CD8 T cells that are persisting up to 75 years post-vaccination. Again, we'll talk about some cool data that came out of this paper a little bit later. There are some caveats to these studies. You might imagine that if we're looking at what happened to you 75 years post-vaccination, you're probably rather elderly, and so we also have to think about effects of what's going on in immune systems of elderly individuals, and these are complicated studies. But the moral of the story is that memory cells can live for a long time. And memory cells also have a number of different unique features compared to some of the other cell types that we're talking about, like the naive or effector cells. On the left, you can see this figure from your textbook that again looks at the levels of different proteins between naive, memory, and effector cells. And you can see that they can be distinguished by combinations of different proteins. That means that we can use our flow cytometer to tell whether a particular cell, these figures are really talking about T cells, but you could do it with T cells or B cells, to tell you whether that cell is a naive, effector, or memory cell. Notice, for example, that the memory cells don't make as much granzyme B because they aren't as necessarily good at killing pathogens, but those memory cells make a ton of BCL2 in order to survive. They also make a ton of cytokine receptor so that they can get lots of signals from cytokines. We can also even see that there are unique subtypes of memory cells like central memory or effector memory cells that will go to different parts of the body that have sort of different resemblances to how much they're like effector versus how other types of cells. And so we've realized that, again, among the memory cells, we don't have a totally homogeneous population. But I want you to also realize a couple of other differences between our naive and our memory cells. So if we look at the genes that are really important for T cells to make or to make proteins of interest to sort of do their function, we find that the chromatin is really closed. We're seeing very little of our signaling proteins and transcription factors being made. And so in order to get transcription of necessary genes, these naive T cells require a lot of signaling. And we've seen this before. We need signal 1, and we need it to actually go for a while, and so the cells have to be like held together with integrins and stuff. We need signal 2, we need signal 3. We really have to hit that cell hard in order to get necessary genes transcribed, to open up this chromatin, to get this DNA off of these histones, to let things move forward. Memory cells, however, have that chromatin really open. The DNA is really accessible. And some of the signaling proteins and transcription factors are already made, so that those cells are going to be triggered much more easily. These cells are generally going to be making a lot more cytokines to help themselves survive, and we're not going to need to hit these cells as hard in order to stimulate them. So once a cell has been activated, if that cell is staying around, this is actually also true of an effector cell. Once that cell is getting activated a second time, it's maybe an effector cell seeing antigen the second time in the infected tissue. So now we're going to be a CTL killing as compared to a naive CD8 cell. Or if we are a memory CD8 T cell, that cell that's been activated before now no longer needs such heavy stimulation to get it turned on. That chromatin is open. We don't need to hit that cell as hard to turn it on. It's already been given its pat on the head and told that it was useful once, so it's not going to be as hard to turn it on. That cell does not need CD28 co-stimulation. We don't need signal 2 anymore. Signal 2 is really important the first time a T cell is activated when it's a naive cell, but the second time around and after that, the cell does not need CD28 anymore. This is going to be something that's going to come up when we talk about peripheral tolerance later, so you can sort of note that here. And because those cells can be so easily activated, they're going to be able to respond very quickly and divide a lot of times in response to our antigen in order to make this nice fast secondary response compared to the slower primary response. What you can also notice is that some of our types of memory cells circulate in lymphoid organs, like the lymph node or spleen. And so we don't have all of the need for as much trafficking this time around. And so all of those things that were giving us a delay in the adaptive immune response in the primary response are not really true in the secondary immune response. These cells get triggered really fast. They're already in the right place. And when they get triggered, they divide and divide and divide and divide and divide and divide because they already had most of the things they needed ready to go. And they were already making cytokines. So they were kind of pre-active. And that allows us to make this type of immune response called a secondary immune response that is considerably more potent than a primary immune response. So here you can see some details of the differences between primary and secondary immune response. At the primary response, we have a really small number of pathogen-specific cells at the start, while in the secondary response, we have a larger number. So some of it's just numerical improvement. In the primary response, we've got this delay that we didn't have in the secondary response. In the primary response, the cells have a high threshold to be activated. The cells have a lower threshold to be activated in the secondary response. We have that delay, like we said, in the primary. We have the cells that are ready sort of immediately and can start acting in a secondary response. If we're thinking specifically about B cells, remember that in the primary response, we have not yet gone through the germinal center reaction, and so we do not yet have class-switched antibodies. We also have kind of this mixture of affinities, so we might have kind of some IgM that might not be useful against the pathogen, and it might only have kind of meh affinity. Whereas by the secondary response, we've had those B cells get time to undergo the germinal center reaction. And so now we have class-switched B cells that are going to make the ideal type of response. And we're also going to have high affinity antibodies for that pathogen because we have undergone somatic hypermutation. Primary responses are, of course, also happening after innate immune responses. And so the innate and adaptive response can't collaborate as much. While in a secondary response, that adaptive response is happening at the same time as the innate response, and so the two types of responses can collaborate quite a bit. On this slide and a few of the next couple of slides, we're going to look at some differences in primary and secondary responses, specifically thinking about an antibody response. And so we can first see on the left the first time that one is infected or first time one is exposed to an antigen versus the second time. In the black line, we see the total antibody response. And so you can see our total antibody response shown here, showing our primary response, and then you can see our total response with the secondary response. One thing that you can notice is that in the beginning, the entire total response is being made by IgM, which is shown in green. Eventually, we start to get some other isotypes. So eventually, IgM is not the whole total. Eventually, we start to get mostly a good amount of IgG because of class switching. And so the sort of unfilled area might be IgE or IgA. But you can see that getting some of those other non-IgM isotypes is going to take us a little while. Then what you can see is that when we have the secondary response, we're never going to have just IgM. We've got some IgM, we've got some IgG, and you can see that we make a massive amount of IgG and predominantly IgG compared to previously when we were making IgM because of those cells that have undergone the germinal center reaction. And you can also see that those other isotypes are being made quite a bit. On the right, you see figures from your textbook with the first, second, or third time one is seeing antigen. On the top, you can see antibody amounts. So you can see IgM in green and IgG in blue. And so at the beginning, we make some IgG, and then we'll start making IgM. But in second and later times that we see antigen, the antibody amount goes up pretty dramatically. And that is largely because of the IgG. If we look at the affinity or how strong the binding is of the antibodies for antigen, you see that the IgMs get a little better, but not a ton over time. But the IgGs with different responses, secondary and so on, really increase pretty dramatically in their affinity thanks to that germinal center reaction. So you can see that if we compare primary and secondary antibody responses, in the secondary response, we have B cells that are specific for our antigen of interest that are way more frequent. We've got high affinity, we've had lots of somatic hypermutation, and we will be getting only the class switched antibodies, particularly IgG. Remember, IgG is by far the most predominant antibody, particularly in the blood. And so that's why we tend to just look at the IgG levels, because we're not missing too much if we're only looking at IgG levels. And you can see this table kind of doing a similar comparison and contrast. So you can see that the secondary response is larger than the primary response. The primary response is usually mostly IgM, while the secondary is mostly IgG. You can see that in the primary response, we have lower affinity, whereas in the secondary response, we have higher affinity because we've undergone affinity maturation. But notice that all these amazing benefits that we see during a secondary response are only possible for protein antigens, because we need to have T cell help to allow our B cells to make those T-dependent responses that include our all of the responses we saw with the germinal center reaction last time that have all of these benefits of the secondary response. We can also see this in the importance of the enzyme AID in these processes. So we can look at mice that either have AID, and yes, these are mice that are actually heterozygous, so they have only one copy, versus mice who have no copies of AID. And we can look at how much IgM they make in response to an immunization and how much IgG they make in response. So the first we'll just look at the solid circles, which are the mice that have AID. And you can see that if we look at how much antibody they make in response to the first immunization, they make some, and it kind of peaks and then goes down. And you can see with the second immunization, they make a little more. But that is, and this is in particular with IgM, you can see that looks pretty similar with AID. Deficient mice, which are in the open circles, because, well, you don't need to have AID to class switch or do hypermutation. So things aren't going to change a whole heck of a lot. But if we look at the IgG response, look at how much of an increase we have over time and as we have in second immunizations in these mice, and also look at how much this just never happens in an AID deficient animal. This slide and the next slide are very T cell specific slides. However, again, the same kind of phenomena have been observed with B cells. It's just that this slide is a T cell slide. So I've mentioned before that that memory cells are not a homogeneous group. And so we're learning more and more about the fact that some of them tend to hang out more in secondary lymphoid tissues. Others will hang out in both tissue and secondary lymphoid tissue. And some, the resident memory subset, seem to only hang out in tissues. So you might imagine that perhaps if you had a SARS-CoV-2 antigen or a T cell that was turned on by SARS-CoV-2 in the lung, you might make some memory T cells that just stay in the lung, because that's a good place to find SARS-CoV-2. Those are, as you can see in this figure, those don't ever spend time in the blood. They just spend time in the tissue, while the other two types spend part of their time going through this red cylinder of the blood. That means that we actually didn't realize that these resident memory cells existed for a long time, because we often just look in the blood. We don't grind up your lung to look and see if there are T cells there. And we know far less about those memory cells, but they actually might be the most important ones, as they are the ones that are actually in the tissue and are probably going to be a very good first line of defense. We can distinguish all of these different types of memory cells by different proteins on their cell surface via flow cytometry. And so we can easily tell the difference between naive T cells, effector T cells, and many of the different types of memory T cells. And so often what we do is that we will take T cells out and we'll use complex flow cytometry to determine, are they effector or are they memory cells, and which of the many different types of memory cells are they. So this image kind of gets at that one little picky detail that I mentioned to you before, which is an area in the field that has been a bit of a controversy in terms of effector cells versus memory cells. And so one of the things that we are less certain about is whether naive cells basically have a choice to either become an effector cell or a memory cell, or perhaps maybe all the cells become effector cells and just the ones that actually live through being an effector cell become a memory cell. The data really looks more like when our naive cell gets activated, some of the cells choose to become effector cells while some choose to become memory cells. And so they go down different paths from the beginning, but this does actually show either option somewhat nicely. You can see sort of this little bit of a controversy here. So the figure that I showed you before showed you kind of that all of these cells go as effector cells. Only some survive to become memory cells, while the majority die. While others have this idea that some naive cells might just decide, hey, I'm going to grow up to be a memory cell, while others decide they're going to grow up to be a short-lived effector cell. And there's even this really cool paper that actually says that it may have to do with how the cell sets itself up at the first division following interaction with the antigen presenting cell. One of the models that seems to have gained a lot of support now is that when a cell is first activated, it's sort of more like a stem-like memory cell that has the ability to divide many, many times and make many more copies of itself, maybe because that cell got sort of a little bit less antigen or lower antigen exposure. And then as that cell gets more and more antigen, gets a little bit harder and harder of a signal, that cell will eventually go to an effector, becoming an effector cell, where it will have less ability to self-renew. So in some ways, we think that it actually might be a little bit of the opposite, that the effector cell is kind of the one that got the strongest signal, and we don't really think that they can go back to becoming a memory cell at this point. One other thing that I'll just tell you is that we have realized that memory development, this is a T cell specific slide, is incredibly dependent on many different cytokines. And these cytokines are all members of the common gamma chain cytokine family that we talked about earlier in the semester. So remember that we talked about the importance of IL-2 for clonal expansion and survival of T cells. But other members of this family, like IL-15 and IL-7, can be really important for helping the cell differentiate to become a memory cell and to help that cell survive. You've already seen IL-7 earlier in the semester that was able to help the developing B cells and developing T cells survive in either the thymus or the bone marrow before they had a receptor. So it's really good at giving cells signal to survive. It turns out memory cells are using that exact same cytokine to get a signal that allows them to survive for a really long time. And this is actually how one would do experiments like this. So these are some experiments that I did in graduate school where I was looking at a response to a particular virus, a recombinant adenovirus. I infected the mice at day 0, and those mice either were wild type, which are shown in the purple line, or mice that were missing a cytokine receptor. This is IL-21 receptor, which is another member of the common gamma chain family. And you can see the mice who are deficient, they don't make a great primary response, but they do make a bit of a primary response. It's actually kind of about the same max that it was with the wild type, though there's maybe a bit of a difference in the contraction phase. But what you also can notice is that when those mice are infected a second time, basically, if they don't have those cytokines to help cells know how to become memory cells, then we just get another primary response over and over and over again, instead of getting the nice secondary response that we see in the wild type mice in the purple. So that's kind of just how experiments like this might be done and what this type of data would look like. And so this is kind of going back to what we've seen before with some data about phases of a primary response. So remember that, previously we've talked about those naive cells, their ability to expand, how they differentiate to be able to act against microbes, but know that that differentiation also kind of sets them up to be short-lived and eliminate antigens, but also then undergo apoptosis, and that some of those cells will be memory cells. And we think that's actually part of their differentiation process. So some differentiate into effector cells while others go into being memory cells. Those memory cells are very long-lived cells that have improved properties, both in terms of the quantity that we have and their quality of expansion to be able to make a really strong adaptive response. One of the other things that I think has been really interesting to me about this process is that we have started to learn a number of things about this process and about some ways that some pieces of it can be sort of complicated during the SARS-CoV-2 pandemic, both in terms of looking at people's responses to infection or more commonly looking at people's responses to vaccination. So there are papers upon papers upon papers that have been looking at these types of looking at some data that is related to stuff we're talking about right now in the class, and that has really helped us to learn some new things. And many of the places that we're learning more about all of this actually tie into some details of the germinal center reaction that we talked about last time, and sort of what's going on with our B cells as those B cells are activated and differentiate into either effector cells like the plasma cell we've seen or into memory cells, as well as what's going on with their somatic hypermutation and their class switch, and how many times that cell kind of repeats going through light zone, dark zone, light zone, dark zone, through the germinal center reaction before leaving as either a plasma cell or a memory B cell. So just to show you a little bit of data that is sort of interesting about all of this. These are the data that I've shown you before, earlier in this lecture, looking at antibody levels against SARS-CoV-2 following infection. And so again, we see data from patients either 1.3, 6.2, or 12 months after infection, and we're looking at two different antigens, and we can see that the antibody levels decline over time following infection. You might say, if you were kind of trying to give a very simple description of this, that the antibody response got worse over time. And in some ways, you might look at this and say, yes, this is what we expect because of the contraction phase. But there are other ways that someone might measure or try to think about the antibody response over time. Here, you can see the number of memory B cells over time in a number of individuals. And what you can notice is that the memory B cells increase over time and then kind of get to this peak number and stay there. And so while in the first, say, 12 months, you could say, well, we have this decrease in antibody levels, we also have gotten this nice increase in memory B cell levels, the percent of B cells that are memory B cells. And so some people might say that's better. That's an improvement, while people might look at these data and say, oh, the response has gotten worse. So whether your response is worse or better kind of depends. Are you talking about amount of antibodies or amount of memory B cells? We can also look at the data based on some of the things that we see here. So in the data that you see here, you can notice a couple of different things. So in the panel, I'm actually first going to talk about this panel that's shown in the middle. Here you can see antibody levels, and these are over time. And here we're looking at people who got vaccine doses either 1.3 or 5 months ago, or then someone who got another vaccine dose a month later. And yes, these are second shots and third shots of the SARS-CoV-2 vaccine. And we can look at antibody levels against the particular strain of the virus, so the exact same genetic type of the virus that the patients were vaccinated with. And you can see that antibody responses go down over time, sad. When we boost and give that additional dose, they go back up, and they go way back up. I mean, this is like, look at the numbers for antibody levels. It's not a small increase. And so that's, partially telling us how potent those memory cells are. But we could also look at the binding strength, or really this is effective concentration. So how good are those antibodies at doing their job? If you need less antibodies to do the job, that means the antibodies are better at their job. If you need a lower concentration to do something, then the antibodies are working better. And what you can notice is that over time, as you get later in the response, the antibodies, while there might be fewer of them, like we saw here, there are fewer, those antibodies are actually better at their job. They have a better amount of binding. So have the antibodies at 12 months gotten better, which is what we might see here because they have a better response and a stronger response, or have they gotten worse because we have fewer of them? Well, what you might realize is that this is probably telling us that over time, those cells have the ability to go through the germinal center reaction, do more somatic hypermutation, and thus get this better antibody response. We've also realized that there's something else that happens with this additional ability to have some somatic hypermutation, and this is related to the data that is in this sort of middle of this slide and in the data on the right that I'm going to show you. So when everyone got their original SARS-CoV-2 vaccine, that vaccine was against the original sequence before there were mutations, before there were variants. It's known as Wuhan Hu1. And so you can see the antibody levels in people who were vaccinated with a vaccine that was made against Wuhan Hu1 after 2 doses over time, but then after that third dose. But what's really cool is if we look after those first couple of doses, we can see that there are some antibodies that bind to other variants of the virus, like Beta, Delta, and Omicron, but they don't bind as well. They're kind of, not so great at their ability to bind, particularly you can see to Omicron after those 2 vaccines. And that makes sense. Remember, the adaptive immune response is really specific. And so if you got a vaccine against the Wuhan Hu1 virus, then you shouldn't have an amazing antibody response to Omicron. But what you can also see is that as time goes on and as we're seeing those memory cells make a response from that third vaccine dose, suddenly we're starting to get a really good response even to unmatched antigen. Or antigen that's sort of closely matched but not quite exactly the same. And you can see now, suddenly after the boost, we get some responses to Omicron, we get some responses to Delta, we get some responses to Beta, even though those weren't in the vaccine. And we've actually realized that this is also something that's happening because of how long somatic hypermutation is actually going on, how long the germinal center reaction's going. This response might be going a little longer than we thought. So maybe when we draw the length of some of these responses, we should think about this antibody response and this germinal center reaction going for a bit longer, and that impacting a bit of what we see for that secondary response. And so that will very likely inform a lot of our vaccine recommendations and our vaccine development in the future, which I think is pretty cool and is one of many things we've learned. And so we'll be talking about some of these details with kinetics and memory, both with a paper that we talk about next week, as well as in the practical immunology assignment that you'll have for next week. On Friday, we're going to talk about NK cells. Thanks for putting up with another video lecture. I will see you on Friday, and I hope that you all have a great next couple of days.